calcium than in milk. So, அப்போ முட்டை, கேரட்டு, spinach, கீரைகள் and egg, fruits. சேர்ந்ததுதான் ஒரே மரம், முருங்கை மரம். All different vegetable, different fruits and egg and milk also. So these all are things equal to MEDICINAL. So good for health, but all are ignored, all youngsters. So especially ladies, so all vitamin E, vitamin D, and also iron content. So another slide, the same moringa oleifera. So different parts of the tree, பூவு, இலை, தண்டு, petioles and roots. So vitamin, iron, calcium, root it consists of iron, zinc, and vitamin A, C, E, and all those things. Anti-diabetic, anti-oxidative, anti-obesity, anti-cholesterol, anti-inflammatory, etc. All list of the disease through the moringa leaves, moringa flowers, moringa stems, stem juice. ஒண்ணு முடியாது. வாரத்துல 2 நாள் முருங்கைக்கீரையும் கொஞ்சம் பூண்டு, கொஞ்சம் மிளகு, உப்பு ஒரு குக்கர்ல 2 கைப்பிடி கீரை, ஒரு 4 டம்ளர் தண்ணி. ஸோ இதை குக் பண்ணீங்கன்னா 2 or 3 விசில்ஸ் பண்ணி குடிங்க. கொரோனாவே வராது உங்களுக்கு. புளிச்சை கீரை எடுத்துக்கணும். இந்த 2 கீரையும் மற்ற அரைக்கீரை மற்ற கீரைலாம் வீக்லி ஒன்ஸ் எடுத்துக்கணும். அட்லீஸ்ட் வீக்லி 3 டைம்ஸ் கீரைகள் சாப்பிட்டீங்கன்னா குட் ஃபார் ஹெல்த். எஸ்பெஷலி முருங்கைக்கீரை அண்ட் புளிச்சைக்கீரை. அது ஹைபிஸ்கஸ் எஸ்குலென்டஸ் புளிச்சைக்கீரை. ஸோ இந்த கீரைகள் ஒழுங்கா சாப்பிட்டீங்கன்னா தென் குட் ஃபார் ஹெல்த். இட் இஸ் ஆல்சோ த பயோகெமிஸ்ட்ரி. வாட் ஆர் த காம்போனன்ட், வாட் ஆர் த என்சைம்ஸ் பிரசன்ட், வாட் ஆர் த விட்டமின்ஸ் ஆர் பிரசன்ட். Vitamin E, tocopherol, and vitamin C, ascorbic acid. Vitamin B1 is a thiamine, and B2 is a riboflavin. So B6 is a niacin, B12 is a copolamine. These all are the secondary metabolites are present. All health benefited vitamin compound, water soluble vitamin and fat soluble vitamins from source of the plant material, especially in முருங்கைக்கீரை. So ஒவ்வொரு part of the moringa oleifera, each and every part of the organ is most important for good for health. It is also one among the medicinal plant. So another plant, terminally arjuna, is a neer marudu. இதனுடைய bark தான் very very important for medicinal value. இதனுடைய பார்க்கு இதை பார்த்தீங்கன்னா இந்த இங்கேருந்து நம்ம சென்னையிலேருந்து டெல்லி போகும்போது ட்ரெயின் ட்ராக்ல தமிழ்நாடு எக்ஸ்பிரஸ்ல போனீங்கன்னா தெரியும், இல்லைன்னா ஜிடியில் போனீங்கன்னா தெரியும். 2 பக்கமும் ஃபுல்லாக நீர் மருதா இருக்கும். ரயில்வே ட்ராக் பக்கம் ரோட்ல கூட கிடைக்காது. நீர் மருது. இந்த மரத்தினுடைய பட்டைய bark is very important for medicinal value. It consists of all secondary metabolites, the terminally arginine, the compound is secondary metabolite, arjuna tonic or arjuna capsule to prevent hypertension, BP, sugar. The powder 1 teaspoon, put it in your buttermilk per day 1 time or twice to control your hypertension. So otherwise the capsule 250 mg capsule or 500 mg capsule or 100 mg capsule, based on that the bark to control your hypertension. So another one is a very good interesting, the plant Andrographis paniculata. It is belonging to family Acanthaceae. In Tamil Syria Nangai, in English, king of bitter. இது ஒரு லீஃபை வந்து சாப்பிட்டீங்கன்னா அடி வைத்த பெறட்டும். அந்த அளவுக்கு இஸ் அ கிங் ஆஃப் பிட்டர், கசப்பு தன்மை உடையது. ஸோ இன் வில்லேஜில் பார்த்தீங்கன்னா இது 1 or 2 லீவ்ஸ், இந்த பாம்பு கடிச்ச உடனே 1 or 2 லீவ்ஸ் புட்டுட்டு இந்த பீட்டல் லீஃப் வெத்தலையில வச்சு மடிச்சு கொடுப்பாங்க டு கண்ட்ரோல் த ஆன்டிடோட். இட் இஸ் ஆன்டிடோட். And also rabies virus also to control. Andrographite, androglapidin, all secondary metabolites. How to extract from the polar solvent and non-polar solvent, dipole solvent based on the solvent system, temperature system, the extraction of the compound from the medicinal plant. The active ingredient is the andrographite, that compound present in the Andrographis paniculata plant. இதுவும் கசக்கும். கீழாநெல்லி, அடுத்து கீழாநெல்லி சொல்ல, அதுவும் கசக்கும். நீமும் கசக்கும். எல்லாமே கசப்பு தன்மை. ஆனா இதை நீங்க பவுடர் ஆக்கிட்டீங்கன்னா எல்லாமே கசக்கும். ஆனா ஐடென்டிஃபிகேஷன், we are educated but very difficult to identification of powder. In the powder form, very difficult to identification of. This is for kீழாநெல்லி, this is for Syria Nangai, this is for neem. Very difficult. But 3 different plant, 3 different application. But one property is a better taste. Every identification, go for phytochemical HPTLC analysis to find out the chemical. Let us know about this is for kீழாநெல்லி, this is for andrographis, this is for neem. And also the identification, this is for a phytochemical analysis. Go for powder analysis for pharmacognosy, powder microscopic analysis, find out the species. Nowadays is a barcoding analysis, DNA barcoding analysis, identification of the species. So the herbal adulteration and substitution also we are challenging also. It is also, as a biochemistry student, you must know about the pharmacognostic methodology, phytochemistry methodology, then another one is a molecular biology, that is identification of the species, validation of the species. So nowadays is a barcoding analysis, the earlier method taxonomical method, then later chemotaxonomical, numerical taxonomical, then phytochemical, then go for right now is a molecular taxonomy, that is barcoding analysis, the identification, because all are இப்போ இது நடந்தது. ஏன்னா கீழாநெல்லியை ஒருத்தரை சாப்பிட சொன்னேன், அவர் கீழாநெல்லி கிடைக்காம வேப்பலையை காய வச்சு அந்த பவுடரை கொடுத்துட்டாங்க. So that powder also it not cure the hepatitis B virus. So automatically they suffered a lot of problem, and also bilirubin content கம்மியா இருக்கும். ஸோ எல்லோ ஃபீவர், ஜாண்டிஸ் வந்ததுன்னா, அந்த ஜாண்டிஸ் வந்ததுன்னா அது நிறைய பேர் வேப்பிலையை பிடிச்சி காய வச்சு நல்லா அதை பவுடர் ஆக்கிட்டு அதை கொடுத்தீங்கன்னா அதுவும் கசக்கும் தான். சார் கசக்குப்பு தான் சொன்னீங்க, ஆனால் அது கீழாநெல்லியா இல்லை சிரியா நங்கையா அல்லது நமக்கு வேப்பிலையான்னு தெரியாது. ஸோ கோ ஃபார் மார்க். அதுக்கு தான் என்னன்னா நம்ம கண்ணெதிரிக்கே வந்து we are cultivated. So to collect the correct species, then go for the treatment. Otherwise, we are going to purchase from the herbal center, some adulteration is there. அதுக்காக தான் அதை சொல்ல வரேன். So it is an anti-inflammatory, antibacterial, antimicrobial. So simply we can say எந்த ஒரு விஷக்கடிக்கும் சிரியா நங்கை is a better medicine. So அதில் பயோஆக்டிவ் காம்பவுண்ட் என்ன காம்பவுண்ட்னா andrographite. So we need to cultivate. ஸோ நாங்கள் பிஹெச்டி பண்ணும்போது இதை கல்டிவேட் பண்ணோம். எங்கன்னா மதுரவாயிலில் 15 acres, மெட்ராஸ் யூனிவர்சிட்டி ஸ்டில் தேர். த கல்டிவேஷன் ஆஃப் மெடிசினல் பிளான்ட் இஸ் மோர் இம்பார்ட்டன்ட். வைல்டு வெரைட்டியும் கல்டிவேட்டட் வெரைட்டியும் மோர் ஆர் லெஸ் ஓகே. ஹைபிரிடைசேஷன் அண்ட் GM crop is I'm not guarantee, I am not sure it. The hybridization variety and GM crop, it is not good for health. So only cultivated and as well as wild variety good for health. Or medicinal plant or vegetables or fruits or any human consuming plants material. So wild and cultivated variety is good for health. So next one is a curry of papaya. It is a belonging to the family is a Caricaceae family. So everyone aware, but you don't know the value of this plant. Only every one of you aware of fruits. Fruits மட்டும்தான் தெரிஞ்சுக்கும். And a leafல பாருங்க, the enhancing the body immunity, the papaya leaves containing phenolic compound and papain alkaloid. These were nutrition strong antioxidant, which in turn enhance the body immunity. And the combination of the papain, the compound help effective the digesting the essential protein which can cure the digestive disorder. Not only for this immune booster, it has also them take for you can drink a glass of papaya juice, adding little lemon juice. Drink this juice at least 2 to 3 times per day. You can cure the dengue fever. Dengue ஒரு காலத்துல ஒரு 2 years 3 years back பார்த்தீங்கன்னா dengue fever. It is not also the English medicine, allopathy medicine. It is very difficult to get it, and also it is not properly side effect. So go for a papaya leaf juice. It cures the dengue fever. So papain is the secondary metabolites, alkaloid components, or flavonoid component, flavonoid classification of compound, lignin content, saponin content. Those secondary metabolites from the, it is not single compound to target, single compound as well as the other small molecule compound supported molecule to cure the dengue fever or any other human disease. It is a synergetic effect. Herbal is a only not single compound to target. It is a synergetic effect. So we have to examine, evaluate each and every part of the plant body. So what are the chemical content or phytochemical content? So extraction, isolation, purification, characterization of those medicinal compound from the medicinal plants, then active ingredient, quantitative and qualitative analysis to target human diseases or plant diseases or animal diseases. Otherwise, we cannot say that. So this is a biochemistry student. You must know about the identification of the species. Which part of the identification of the species is more medicinally important? So medicinally important for fruit or leaf or petiole or stem or bark or root phloem. So we have to examine then for extraction procedure, solvent procedure, then extraction time and solvent temperature and human temperature, then go for isolation, then purification through the bioanalytical techniques or chromatographic techniques, then go for the antimicrobial and antifungal, anti-cancer, anti-saline culture and etc., etc., etc. So these all are the very, very important. That is how you must aware of the plant first of all. And identification based on the pharmacognosy identification, HPTLC, that is a phytochemical analysis, then molecular basis. Or any botanist to help to identification, go for further investigation of the biochemistry, microbiology, biotechnology, or industrial application, etc. But this plant is for only to cure the dengue fever and other aspect also you have to investigate. Dengue fever மட்டும் கிடையாது. We have to பண்ணதிலே பண்ணாதான் research வந்து ஷைன் ஆக முடியும். இப்போ ரைஸ்ல பாருங்க 200 variety of rice. Still rice research is going on. Still agriculture department, rice research and each and every, so 24 chromosome இப்போ ஒர்க் பண்ணிட்டு இருக்காங்க. genome projectல ஒர்க் பண்ணிட்டு இருக்காங்க. Still rice is going on. There is a 40 years or 50 years rice research is going on. Not 40 years, more than 60 years. Similarly, we need the தெரிஞ்ச plantலே தெரிஞ்சதிலே more application we can take it out. We investigate. So in the aspect இது work ஆகுதுன்னா another aspect we as a youngsters think about the same plant. So different angle of the thinking for different application of the disease also. And next one is a basil leaf or Tulasi, Asimum santum. It's uses for making tea and also refreshment of the and good for health. Idile pathina Krishna Tulasi, Sivan Tulasi, more than 35 variety in India. 35 variety. We climbed through molecular biology. We climbed all over the world in DNA barcoding. Asima more than 15 species we collected from Tamil Nadu, Kerala, and Karnataka. We maintain the herbarium. We maintain the herbal garden. We maintain the herbarium documentation, and also we are isolate the DNA, molecular DNA from 15 species, deposited in the NCBI from US, barcode from Canada, and EMBL accession number and Japan and data bank, gene data bank from the Japan. Everything accession number we got it. It is a look like is a patent for molecular biology. We are first reported all over the world. No one claimed these species other country. It is belong. இப்போ பார்த்தீங்கன்னா நீமோ டர்மரிக் வந்து இப்போ அமெரிக்கன் வந்து இது எங்கள் தான் பேட்டன்ட். இப்போ இந்த ஸ்பீஷிஸ் இட் இஸ் பிலாங்கிங் டு இந்தியா, கர்நாடகா, இன் இந்தியா, கர்நாடகா, தமிழ்நாடு, Kerala. We climbed this species is belonging to the South Indian. So we are first reported in barcoding. More than 15 species out of 35 ஸ்பீஷிஸ்ல நாங்க 15 ஸ்பீஷிஸ் ஒரு பிஹெச்டியை முடிச்சிருக்கோம். இதுக்கு தான் யூஜிசி சார் சொன்னார்ல, யூஜிசி ஃபண்டட். ஸோ ஒய் ஐம் டெல்லிங், ஸோ இந்த இன்ஸ்டிடியூட் இருக்கு, ஒரு இன்ஸ்டிடியூட் CMAPன்னு கேள்விப்பட்டிருப்பீங்க, சென்ட்ரல் இன்ஸ்டிடியூட் ஆஃப் மெடிசன் அண்ட் அரோமேட்டிக் பிளான்ட்ஸ், புரம் நகர் அட் லக்னோ. இட் இஸ் அ CSIR லேப். One more institute, National Botanical Research Institute. It is also Lucknow. It is also the CSIR lab. One more lab, CSIR lab, Lucknow. CDRI, Central Drug Research Institute. Moon institutes. First one is the Central Institute of Medicine and Aromatic Plants. National Botanical Research Institute. Central Drug Research Institute. மூணுமே CSIR lab, எல்லாமே லக்னோவில் இருக்கு. So more than 42 research institute CSIR lab, 3 research lab லக்னோவிலே இருக்கு. So இந்த 3 research instituteலையும், if you are as you are biochemistry student, you go through that website, let you know about what is going on in India. So and one more or 3 more institute is CSIR lab in Kolkata and Delhi also it is available. What is going on the medicinal plant research? They didn't do it for the DNA barcoding from the Asima plant. Nowadays, so we done it more than 8 years before. But right now, every people are doing it. So barcoding analysis. It is one among the species identification. And also that it is belonging to the India. No one claimed. So Asimum santum is there are different applications and human booster. So these all are the information about the Asimum family. Another interesting plant for Trichobus Jylanicus. It is belonging to earlier Trichopodaceae, but recently Dicoraceae family. In Tamil, Arokiya Pachai. This leaf is identified Kerala TBGRI, Tropical Botanical Garden Research Institute. Trivandrum இருக்கு, பாலோடு. ஸோ இது ஒரு லீஃப் ட்ரைபல் பீப்புள் கிட்ட இவர் டாக்டர் புஷ்பாங் CMAP டைரக்டராக இருந்தார். CMAP டைரக்டராக இருந்தார், NBI டைரக்டராக இருந்தார், TBGRIல டைரக்டராக இருந்தார். ஹி ஐடென்டிஃபைடு திஸ் ஸ்பீஷிஸ். அவர் எங்கன்னா கேரளாவில் ட்ரைபல் பீப்புள் கூட மிங்கிள் ஆனார். ஸோ அப்போ ட்ரைபல் பீப்புள் வந்து காலையில் மார்னிங் பிரேக்ஃபாஸ்ட் 1 or 2 leaves. It is a immune booster and good food. It is equivalent to 4 idli or dosa, etc. 1 or 2 leaf is good for health. Morning breakfast is 1 or 2 leaves. It's good for health. Arokiya Pachai idukku peru. So we need more research in this plant also. It is only available in southern part in Kerala, in Palod, TBGRI. I don't know where it is. The biodiversity of Trichobus Jylanicus அல்லது ஆரோக்கிய பச்சை. So if you are interested, it is where you go investigate more. It is a lacking of research also. Only the antimicrobial, antifungal, known try for anti-cancer activity also. So if you are interested, please you take this plant from that area, continue your further future research also, novel compound or innovative investigations, and real story you can find out. It is a boosting for leaves and fruits used for medicine. People take Trichobus Jylanicus for improving the stamina, boosting the immune system. So losing our weight. நல்லா obesity போட்டுறாங்களே, லாக்டவுன்ல பார்த்தீங்கன்னா நிறைய பேர் obesity. So losing your weight. They also take it to treat liver disease, stomach, ulcer, and sexual performance and urinary infections, etc. So another one is a Philanthus Amaris. Don't call in India this plant is, don't call Philanthus niruri. Nirurin கூப்பிடாதீங்க. It is a niruri is a West Indian species. Philanthus Amaris is a Indian species. So World report said that Lancet journal, medical journal, are you aware of it? As a biochemistry student, you are aware of Lancet journal, medical journal. So I reported this plant, SPT Tyagarajan and other, the Europe and American authors said that Philanthus Amaris, only South Indian species, it cure the jaundice disease. World report said that. I'm not saying that. உலகத்திலே வந்து சவுத் இந்தியா தான் கீழாநெல்லி மட்டும்தான் குணப்படுத்துது. மஞ்சக்காமாலை, ஹெப்பாடைட்டிஸ் பி வைரஸ். அப்ப எவ்வளவு வேல்யூபிள். அப்ப நார்த் இந்தியாவிலையும் இருக்கு, அக்ராஸ் த வேர்ல்டு எங்க வேணாலும் இருக்கு. ஆனா அது குணப்படுத்தல. இப்ப ரிப்போர்ட் வந்து சவுத் இந்தியாவில் இருக்கு, அது எஸ்பெஷலி இன் தமிழ்நாடு பிளான்ட். தமிழ்நாடு அப்ப மண் வளம் எவ்வளவு நல்லா இருக்கு பாருங்க. தட் இஸ் அ டைவர்சிட்டி ஆஃப் த ஸ்பீஷிஸ். இங்க இருக்கிற diversity of species, same species in other country also, it won't cure the same disease. So that is the more important for the soil factor, environmental factor, then soil analysis, chemical analysis, pH, soil properties, then atmosphere, then find out the value of the species and particular area, the more applications, more curable the disease. So good example கீழாநெல்லி. ஸோ தமிழ்நாட்டில் இருக்கிற கீழாநெல்லி தான் லான்செட் ஜேர்னல்ல இருக்கு. இத வேர்ல்டு ரிப்போர்ட்லேயே சொல்றாங்க. கீழாநெல்லி அதுவும் தமிழ்நாட்டில் இருக்கிற கீழாநெல்லி தான். இதுதான் இப்போ நான் சொன்னேன். இப்போ இந்த கசப்புத்தன்மை பவுடரா மார்க்கெட்ல விற்கும்போது வேப்ப ஈஸியாக கிடைக்குது, அதை வந்து பவுடர் ஆக்கி கொடுத்துட்றாங்க. ஸோ இட் இஸ் வெரி tough task for we are educated, very difficult to find out the powder form. Go for analysis, pharmacognosy analysis, phytochemical, and then molecular biology analysis, find out the species. Always very tough task. So Kiranelli is a Philanthus Amaris. Don't call கூப்பிடாதீங்க. இதுக்கு பேர் வந்து Amaris மட்டும்தான். நிரூரின்றது வெஸ்ட் இண்டியன் ஸ்பீஷ். ஆனா மோஸ்ட் ஆஃப் த ஜேர்னல் நிரூரின்னு போடுறாங்க. ராங் ஐடென்டிபிகேஷன், மிஸ்நார்மல்ஸ். Keep in your mind, please ask youngsters, all old people also, wrong identification. Niruri inga kediyadu, Indiavile kediyadu, West Indian species. So Philanthus Amaris. Tamil la sollana Keelanelli, Sanskrit la sollana Bhoomiya Malaki. So it is a cure for the idhula philanthine compound and hypophilanthine compound. In the plant தான் என்னுடைய PhD work, cultivation of herbal plant. ஆயுஷ்ன்னு சொல்றோம்ல, இப்போ ஆயுஷ் என்னன்னா ஆயுர்வேதா, யூஃபயா, யங் அண்ட் நேச்சுரோபதி, யுனானி, சித்தா and homeopathy. The earlier name before 2014, before Modi government, it is a Ministry of ISM and Homeo, Indian System of Medicine, Homeo. Now change Ministry of Ayush. So that kind of project, under the project I worked, I got the PhD from the medicinal plant, especially in Kiranelli, cultivation, phytochemistry, and pharmacognosy. I got a degree from the Madras University in CAS in Botany, Philanthus Amaris. So I cultivated more than 50 acres across India, especially in Tamil Nadu. So in southern part and the western part, I cultivated and lively cultivated during my PhD period. I succeed and I published in Navina Velanmai. I succeed published in the paper also. 2000 Navina Velanmai. Navina Murayil Kiranelli Valapur. Okay. So this is why I'm telling phylantine and hypophyllantine. Without hypophyllantine and phylantine compound, it won't cure the jaundice. Phylantine and hypophyllantine mattum cure பண்ணாது. So அந்த compound வந்து எந்த compound இருக்குதோ அந்த compound. Okay. The next one is Cissus quadrangularis parandi. It is a cure for the good for health digestion system. It is a carotenoid, triterpenoid, ascorbic acid, and also it is cure anti. It is a good heavy meals எடுத்தீங்கன்னா பரண்ட துவையல் சாப்பிட்டீங்கன்னா it's good for digestion tablet but digestion நிறைய பேர் சாப்பிட்றாங்க சார் ஜீரணம் ஆகலை பரண்ட துவையல் சாப்பிடுங்க சரியாயிடும் because all are secondary metabolites good for health, all primary metabolites good for health. The next one is Catharindus rosea. Are you able to see my slide?
Yes, sir.
Okay. So read it. This slide, read it, ma.
Catharindus rosea is commonly known as.
Okay. Thank you.
Yes, sir.
Yes,
sir. Visible. No problem.
Okay. Catharindus rosea. It is a belonging where that is a Apocynaceae family. Catharindus rosea. It is a Sudugattu Malli in Tamil. Sudugattu Malli. Nowadays, the collected from the raw material from India, the manufacturing the tablet for cancer tablet, vincristine, vinblastine, those active ingredient are phytochemical constituent isolated from the வெள்ளை and blue. சுடுகாட்டு மல்லியிலிருந்து ஐசோலேட் பண்ணி அதை ப்ராடக்டா இம்போர்ட் பண்றாங்க மெடிசன்ஸ். இன்னைக்கு கொரோனா வச்சு நிறைய கேம் விளையாடுறாங்க. அதெல்லாம் இட்ஸ் அ பொலிட்டிக்கல் கேம் ஃபார் வேக்சின் ப்ரொடக்ஷன்ஸ். Similarly, in the same raw material from India, but manufacturing component and the patent of those components from the America. Imported, அதுக்கு எல்லா வரியும் போட்டு ஜிஎஸ்டி, சென்ட்ரல் ஜிஎஸ்டி and state GST, we get the import. இன்னைக்கு வந்து anti-cancer, different form of the cancer. It is used to treat the various form of the cancer. Uterus cancer, breast cancer, and liver cancer. There are so many cancer in human body. It cures the vinblastine and vincristine. Tablet பார்த்தீங்கன்னா cancer patientல பார்த்தீங்கன்னா அதுல இருக்கும் பின்னாடி vincristine, vinblastine. Those compound isolated from the Catharindus rosea. That is Sudugattu Malli. It's a belonging Apocynaceae. Please you keep in your mind all those medicinal plants. We need to patent at least Indian patent. காப்புரிமை வாங்க வேண்டும். Intellectual property rights. ஒரு லாயரை IPR லாயரை பார்த்து you can talk to any novel compound or any novel applications or industrial applications from the plant source. So more application industrial or social oriented, definitely go for the patent copyright. That is the most important. எல்லாமே பண்றாங்க ஆனா பேட்டன்ட் போறது இல்லை யாருமே நம்பாளுங்க. So that is a lacking. So after getting publication, then go for the submission of the thesis and get the award PhD. அந்த மோட்டோல தான் இருக்காங்களோ வச்சு no one social oriented, society oriented, commercial oriented, think about the long vision, any one plan. So think about your isolation, extraction, purification, characterization applications for single medicinal plant. That is the most important. So here itself the vincristine, vinblastine I said. So next one is a very good interesting plant. Nowadays we are facing a lot of problem young couples. ஸோ இளமை துடிப்புடன் வாழ ஓர் இதழ் தாமரை. வரப்புல பார்த்தீங்கன்னா நம்ம வில்லேஜ்ல போனீங்கன்னா வரப்புல எல்லாம் இதெல்லாம் இருக்கும். ஓர் இதழ் தாமரை பார்த்தீங்கன்னா ஒரே ஒரு பெட்டல்ஸ் இருக்கும். ஸோ குட் ஃபார் ஓர் இதழ் தாமரை. பெஸ்ட் ஒன். இன்க்ரீசிங் த ஸ்பெர்ம் கவுண்டிங் அண்ட் ஆல்சோ லெபிடோ. பால் இணை. இப்போ நிறைய டிவியில பார்த்தீங்கன்னா உங்களுக்கு சார் எனக்கு இப்போ நிறைய ஐவிஎஃப் எங்க பாரு ஐவிஎஃப் இருக்கு. ஐவிஎஃப்னா இன்விட்ரோ ஃபெர்டிலைசேஷன். இன்விட்ரோ ஃபெர்டிலைசேஷன் ஃபார் சரோகேட் மதர்லாம் வந்துச்சு. பிகாஸ் ஆஃப் அவர் ஸ்ட்ரெஸ் மேனேஜ்மெண்ட். ஸ்ட்ரெஸ் ஃபார் ஏபயாட்டிக் ஸ்ட்ரெஸ் ஆர் பயாட்டிக் ஸ்ட்ரெஸ் ஆட்டோமேட்டிக்லி suppress and male sterility, female sterility, fibroids, etc. These all are the cause. Means our lifestyle is changing. No one bother about our traditional food. No one bother about our traditional material, plant materials. So everyone fascinated to go look for our KFC, McDonald's, and pizza. It is all those food material not good for health. Automatically male sterility, female sterility, or fibroid without fertilization properly. The young couple, the malady innuvaanga. நிறைய வீட்டில் இப்போ பிரச்சனை கோர்ட்டு கேஸு அப்படின்னு தான். We get proper medicine from the natural resource, definitely good for issues. Always very well. The sperm இருக்கும் நிறைய பேருக்கு sperm ஒரு rejection sperm but not viable. ஸோ அப்போ என்ன பண்ணுவாங்க, நீங்கள் ஐவிஎஃப் போனீங்கன்னா ஐவிஎஃப்ல லட்சக்கணக்கில் செலவு பண்ணி வச்சு அங்கே நடக்கிற திலும்புல யாருமே வெளியில சொல்ல மாட்டாங்க. The sperm collected from the other person also. So and uterus also there, then for IVF in vitro fertilization அந்த கருமுட்டியை எடுத்து then go for அந்த பெண்மணிக்கே வைக்க முடியாத நிலைமையில எல்லாம் இருக்கு இப்போ. பிகாஸ் ஆஃப் த malnutritions, genetic character. ஸோ இப்போ இதெல்லாம் ரீசெண்டாக நடக்குது. எங்கன்னா பஞ்சாப்ல நடக்குது. ஸோ இப்போ சரோகேட் மதர், வாடகை தாய், சரோகேட் மதர் we are going to look for them. அப்போ அந்த கருமுட்டி இந்த ஜென்ஸுக்கு இந்த ஸ்பெர்ம் உடைய வயபிலிட்டியும் இல்லை, அந்த கர்ப்பப்பையில அந்த கருமுட்டியை வைக்கிறதுக்கு தகுதியும் இல்லை. ஸோ வி ஆர் லுக் ஃபார் த அனதர் லேடி. ஸோ தட் இஸ் ஆல்சோ ப்ராப்ளமேட்டிக். ஸோ ஆஃப்டர் 10 months தட் லேடி ஸோ கெட் த சைல்டு வந்தோடனே அந்த குழந்தை வந்து யார் சமூகத்தில் நிறைய பிரச்சனை வருது. ஸோ இதெல்லாம் எதுக்கு சொல்ல வரேன்னா நிறைய இஷ்யூஸ் அண்ட் இது நல்லா பேசுறதுக்கு நல்லா இருக்கும், டெக்னாலஜிஸ் ஒரு வெரி குட்னு சொல்லுவோம். ஆனா இதுல லைஃப்ல இருக்கிற ப்ராப்ளம் பாத்தீங்கன்னா பிக் இஷ்யூஸ். இதுக்கெல்லாம் காரணம் ஒழுங்கா ஃபுட்டு எடுக்காதுதான் காரணம். என்னுடைய மெயின் காஸ் இஸ் we are not follow our traditional foods. So that is the most important. So அந்த குழந்தை யார், அப்பா சொல்லும், அம்மா சொல்லும், யார பெற்றவங்களை சொல்லுமா, ஸ்பெர்ம் கொடுத்தவங்களை சொல்லுமா, இல்ல வளர்க்குறவங்களை சொல்லுமா என்றது பிக் கொஸ்டின் மார்க். ஸோ அதுக்காக தான் that is a master. The common name is spat flower. To come to the point, this plant is a suranam. 1 teaspoon of this powder mixed with milk and take in the morning, evening, strengthen the body and help their regaining the lost vitality. So another usage of powder for mixed with black pepper and drink treat urinary disorder. The leaf juice mixed with the goat milk. So the bowl. We are fruit used for treatment of cough in the tail kadi, illana vaitha vali, stomach pain. During your periods, youngsters, especially girls, during periods time the stomach pain இருக்கும். At that time 1 or 2 teaspoon மில்க்ல கலந்து ஆட்டுப்பால்ல கலந்து சாப்பிட்டீங்கன்னா it will cure the abdominal pain also. That is about complaints. And also the Oridai Tamarai Suranam is excellent Siddha medicine. It helps to increasing the libido. Libido means பால் உணர்ச்சிகளை தூண்டக்கூடியது. Improve the quality of semen. That is important, very, very important. Quality of semen is very, very important. And in women, enhance milk segregation after childbirth. So this very, very useful. And also the semen production, சிறுநெருஞ்சி பெருநெருஞ்சி முள்ளுன்னு இருக்கும். That Tribulus terrestris in a botanical name. தமிழ் வந்து சிறுநெருஞ்சி பெருநெருஞ்சி முள். அதனுடைய ஜூஸை குடிச்சா கூட the male sterility So male sterility also to rectify it. So நம்ம இதுல வந்து நிறைய ட்ரெடிஷ்னல் பிளான்ஸ் மூதாதிகள் இந்த ஆயுர்வேதிக் மெடிசன்ஸ் ஒழுங்கா வி ஷுட் ஃபாலோ எல்லாமே ஃப்ரெஷ் மெட்டீரியலா யூஸ் பண்ணணும். அதுதான் பிளான்ட் மெட்டீரியல் வந்து ஃப்ரெஷ் மெட்டீரியலா யூஸ் பண்ணீங்கன்னா குட் ஃபார் ஹெல்த். So it is also the tonic, diuretic, and other properties and antipyretic, anti-inflammatory, etc. All plant is a similar same aspect also. So Nochi, you know that it is also one among the plant Vitaceae family. It is a headache and cure. இப்போ இந்த ஆவி பிடிக்கிறதுக்கெல்லாம் இந்த கொரோனா பீரியட்ல த்ரோட் இன்ஃபெக்ஷன்ஸ் ரெட்டிஃபை இட் இந்த வைட்டஸ் நுகண்டோ இது பென்டாஃபோலியேட் இருக்கு, ட்ரைஃபோலியேட் இருக்கு. வி டோன்ட் ஹேவ் த டைம், எல்லாமே எடுக்கணும்னா கொஞ்சம் கஷ்டம். It is a Vitis nigundo. So another Stevia ribodiana is a chini plant and pere. It is belonging to the Astraceae family. Hyperglycemic. இது வந்து 1 kilo Stevia ribodiana sugar plant, chini plant equivalent to 300 kg of sugar, sucrose. இது வந்து fructose monosaccharide. Fructose வந்து எப்படின்னா galactose குளுக்கோஸும் சேர்ந்துனா சுக்ரோஸா வரும். அது ப்ராசஸ் பண்ணது. ஸோ இட் இஸ் நாட் குட் ஃபார் ஹெல்த் ஃபார் டயபெட்டிக் பேஷண்ட். ஆனால் இட் இஸ் எ குட் ஃபார் டயபெட்டிக் பேஷண்ட். ஸ்டிவியா ரிபோடியானா இட் இஸ் பிலாங்கிங் டு ஆல்ரெடி ஐ செட் த இது அஸ்ட்ரேசி ஃபேமிலி. ஸோ இட் இஸ் ஈக்குவலன் டு ஹவு மெனி டைம்ஸ் 300 டைம்ஸ் ஈக்குவலன் டு சுகர். ஸோ இது வந்து ஆல் ஃபுட் இண்டஸ்ட்ரி, ஆல் herbal industry, all diabetic patient alternate sugar இதுக்கு பேர். Stevia, the benefits and properties, hyperglycemic and main application management, diabetics, adding the weight losses and supporting. It's good for health. If you need more information, please ask through the phone or after the meeting also. Next one is Sauropus androgynous. The Sauropus androgynous is a Tavasi Murungai. It is belonging to family Philanthaceae, Tamil Tavasi Murungai. 1 or 2 leaf equivalent to vitamin B complex. Don't go for Bicosal capsule. Bicosal capsule நிறுத்திட்டு இதில் 2 லீஃப் சாப்பிட்டீங்கன்னா தவசி முருங்கை மெட்ராஸில் நிறைய இடத்துல இருக்கு. இது மதுரவாயில் காருண்ய யூனிவர்சிட்டி, இது மெட்ராஸ் யூனிவர்சிட்டியில மதுரவாயில் கேம்பஸ்ல இருக்கு. ஸோ இந்த 2 லீஃப் இட் இஸ் ஈக்குவலன் டு Bicosal capsule, 1 capsule. So it is good for health. And also the antipyretic, anti-inflammatory, the other human disorders to rectify it. But major is a multivitamin plant. Multivitamin plant or otherwise Sauropus androgynous, Philanthaceae family, all vitamin, green vitamins, all water-soluble vitamins or fat-soluble vitamins equivalent to this, especially in vitamin B complex. Next one, Solana, Kandangatri, good for it is a Kabbalah Sali, it is a cure for the asthma and sore throat infection, toothache, and all high concentration of solanite, starting material, mall, manufacturing, and etc. So another one is the Bacopa moneri. We don't have the time also. The Bacopa moneri, it's a near Brahmi. The another plant is Centella asiatica. It is also memory plus tablet. Vallarai Kirai is a near Brahmi. Rendume vandu improve our IQ as well as memory power. So memory power in terms of the short-term memory, long-term memory, retain the capacity of the memory. So already experimentally done by Dr. Akhilesh Sharma, Ayurvedic expert. Brahmi is excellent for improving your brain. Functioning and strengthening your memory from this Bacopa moneri and also the Centella asiatica. It is a Plantagonaceae family. So another plant is a Solanum trilobatum. It is a Tudu Valley. So it is a leaf. It has thorns. Mulluruko. So it is a Tudu Valley. It is used for the treat common cold, cough. இப்போ இந்த கொரோனா பீரியட்ல தூதுவளை எல்லாம் இப்போ சாப்பிட்டீங்கன்னா த்ரோட் இன்ஃபெக்ஷன் வராது. த்ரோட் இன்ஃபெக்ஷன் வரலன்னா லங்ஸ் அஃபெக்ட் ஆகாது. லங்ஸ் அஃபெக்ட் ஆகுதுன்னா கொரோனாவிலேருந்து ஃப்ரீ ஆயிடலாம். ஸோ அதுக்காக தான் நம்ம வேக்சின் எடுத்தோம்னா, வேக்சின் எடுத்து ஹெர்பலை ஃபார்ம் பண்ணுனா then we can't get any diseases also. So use the treat common cold, cough, asthma, and herb. So consume the mild and praying and oil ghee and with இதை வந்து இது துவையல் பண்ணும்போது கொஞ்சம் கீ விட்டு துவையல் பண்ணீங்கன்னா குட் ஃபார் ஹெல்த். எல்லாம் அந்த கசப்பு தன்மை இருக்கும். அது கூட பூண்டு இஞ்சி போட்டு நல்லா மிக்சியில போட்டு அரைச்சி தூதுவளை துவையல் சாப்பிட்டீங்க. இல்லைன்னா மின்ட்ன்றது புதினா துவையல், இதெல்லாம் ஒழுங்கா சாப்பிட்டீங்கன்னா இட் won't affect the corona. ஓகே. ஸோ நெக்ஸ்ட் ஒன் குருக்குமா லாங்கா மஞ்சள் பொடி. இட் இஸ் a turmeric powder. It is a gingiberaceae family which is belonging to anti-inflammatory, anti-cancer. ஒரு 1 or 2 teaspoon put it in the RO water. ஸோ நம்ம ஸ்ப்ரே பண்ற வாட்டர்ல ஆன்டிசெப்டிக், இட் இஸ் a disinfect. வெளியில வச்சுட்டீங்கன்னா இட் இஸ் more than 1 week or 4 days sufficient for a natural antiseptic. So 1 or 2 teaspoon, put it in the 5-liter water. So and keep it in the outside the house. So whoever come inside the house automatically disinfect. ஸோ இட்ஸ் a natural டிசைனர். நீங்கள் இப்போ இது மாதிரி ட்ரை பண்ணலாம். எதுக்குன்னா ஆல்கஹால் நீங்கள் இப்போ கோவிலில் போனால் எங்கே போனாலும் உங்களுக்கு ஸோ இப்போ அந்த ஸ்ப்ரே பண்ணி உங்களுக்கு கெமிக்கல் அது ஃபுல்லாகவே ஆல்கஹால் ஈஸிலி இமீடியட்லி டு கேட்ச் அ ஃபயர் ஆல்சோ. ஸோ இது பண்ணி பாருங்க. இப்போ டர்மரிக் இல்லைன்னா கிராம்பு, ஸ்பைசி கிராம்பில் கூட பண்ணலாம். நீங்கள் ஒன்றும் இல்லை, ஒரு 5 லிட்டர்ஸ் தண்ணியில் 2 கிராம்பை போட்டுருங்க. ஸோ இது மஞ்சள் பொடி அண்ட் உப்பு போட்டிங்கன்னு காகுல் பண்ணலாம். இல்லை கை காலை கழுவிட்டு உள்ள வந்தீங்கன்னா டெய்லி இதை பண்ணீங்கன்னா, வெளியில நிறைய பேர் வெளியில சுத்திட்டு வரீங்க, இல்லைன்னா நல்லா ஒரு இண்டஸ்ட்ரியில ஒர்க் பண்ணிட்டு வரீங்க, நிறைய பாப்புலேஷன் இருக்கிற இடத்துல இருந்துட்டு வரீங்கன்னா, இந்த 3 கிராம்பு உப்பு மஞ்சத்தூள் போட்ட தண்ணியில காலை கழுவி கையை கழுவிட்டீங்கன்னா, ஸோ அண்ட் காகுல் பண்ணிட்டீங்கன்னா இட் won't affect the any viral disease in your body. So it is a protect yourself and also immune boosters. Aloe Vera, it is a good for health for pulp. It is small amount of turmeric powder is a good remedy for the enlargement of the spleen. It is clean pulp aloe vera jelly. So you can palm jangri. The pinch of asafoetida, பெருங்காயத்தை கொஞ்சம் போட்டீங்கன்னா, the pound them together and store it in airtight container வச்சுக்கிட்டுன்னா, 1/2 gram of the preparation of the consumed twice daily, the easy access of the menstrual pain. So during the girls or ladies, the menstrual cycle நிறைய pain இருக்கும். They are meet out that there are so many issues. So இந்த நீங்க வைத்தியம் பண்ணீங்கன்னா clean palm, the pulp of aloe, palm jangri and pinch of asafoetida இதெல்லாம் சேர்த்து ஒரு together with the store in airtight container வச்சுட்டு 1/2 gram எடுத்து சாப்பிட்டீங்கன்னா it cure for such a problem, menstrual pain problem. 2 teaspoon of the pulp fried in half teaspoon of ghee, and which half teaspoon of the sugar add the consume the help cure and wet cough. So and the body heat also, it is a good for health, and also not only for these all are the day-to-day life, it is also the jelly cure the human tumor also, anti-tumor. The brain tumor it will cure. So already they reported in the medical journals. So Cardiospermum helicobacter is another plant. It is belonging to Sapondaceae family. In Tamil is Modakathan. So Modakathan, dosai Modakathan idli, it is good for health and anti-arthritis and exists significant analysis, anti-inflammatory activity and etc. இது வந்து mucilage form, saponin content அதிகமா இருக்கும் secondary metabolites to control and also the further studies. So next one is the Vitamia swaminifera, for it is belonging to the Solanaceae family. So the Vitamia swaminifera, vithanolide, vitaferin, so all the secondary metabolite is economically important, the medicinally important, root is important, is the brain benefits and also the stamina and anthracitis, improve body defense against disease, improving the cell mediated immunity and against cellular damage causing free radicals. So these all are the control our body and immune boosters. So நரம்பு தளர்ச்சியில் இந்த Parkinson's இதெல்லாம் கண்ட்ரோல் பண்றது வித்தனோலைடு root. The root it comes with vithanolide or vithanolaferin or vithanolaferin A, B, etc. These are all alkali component from the Vitamia swaminifera. Sir, continue பண்ணலாமா எப்படி சார்?
Actually, sir.
பண்ணலாம் சார், no problem.
Dear participants, can we continue? Dear participants, 1 or 2, please answer. How many slides, sir? How many slides are left?
Remaining 30 slides, but conclude பண்ணிட்ட ஒரு 5 to 10 minutes la.
Yes, sir.
No problem, sir, no problem.
We can conclude within 10 minutes, sir.
இல்ல இன்ட்ரெஸ்டிங்கா இருக்கா அதான் கேக்குறேன்.
நல்லா தான் போயிட்டு இருக்கு சார்.
It's going very informative.
Very useful, sir.
Very useful and very informative.
Okay.
You can take your time, sir.
Sorry to interrupt here, sir.
Yes,
It is
sir.
very useful, but we try to give the parts to be used during the preparation.
Okay.
Example, in certain cases, roots may be used.
Yeah, I said for Vitamia somnifera root used, for terminally arjuna bark used, Kiranelli for whole plant used.
Yes. Yes, okay.
And Oridal Tamarai, sir.
What, sir?
Oridal Tamarai.
Oridal Tamarai is a whole plant. Whole
Okay,
plant,
thank you,
sir.
Thank you.
Whole plant is a
Thank
very medicinal value. So it is increasing the viability of the sperm and also libido. Libido means is increasing the பால் உணர்ச்சிகளை தூண்டக்கூடியது. The entire plant.
Okay. As per the feedback of participants, please continue, sir. No problem.
Okay. So now the about the medicinal plant I completed. Then I go for extraction. The medicinal plant is a constituent, the effective source of both traditional and modern medicines. The herbal medicine has shown the genuine utility. So most of in the 80% rural population depends on it. Primary health care is a World Health Organization in 2005, not only 2000, in 2020 also said more than 90% of the rural population depending on the herbal plants. So what are the herbal plants? It consists of phytochemical constituents, that is a weed veli porukal. The natural bioactive compounds found in different parts of the fruits, flowers, stem, leaf, roots, different parts. Most important. Already the participant asked for which part is more important. So each and every part, for example, Kiranelli also, so in plantain compound in leaves is more than in root. Leaf la da adhigama irukum. So in stem is more is the middle. And root is the least philanthine content. Without philanthine, it won't cure the jaundice disease. So that is also very important. The phytochemical component, that component alone not cure the disease. The compound along with other phytochemicals, that is a synergetic effect. All herbal compound synergetic effect, and also whole plant or whole flower or whole stem or whole bark or whole plant. Different parts are whole plant. To make it very clear, they provide the definite physiological action on the human body. Based on the human body, then take for the quantitative analysis for the herbal drug. The bioactive substance including tannin, alkaloids, carbohydrate, terpenoids, steroids, flavonoids, and phenolic component. The widely used in human therapy, veterinary, agriculture, scientific research, and the countless other area have inhibitory the effect of all types of the microorganism in vitro and in vivo. That is the application part. So first of all, let us know the identification. The before slide, the medicinal plant. What are the medicinal plant? What are the different parts of the plant and whole plant? Finally, we are looking the phytochemical. Those phytochemical based on the chemical and body physiological action, then application is going on. So now extraction procedure. There are number of extraction methodology according to the application. The extraction procedure is different. Extraction methodology is different based on the Ayurvedic purpose and the Siddha purpose, Unani purpose, and biochemical purpose and biochemistry purpose and biotechnological purpose. So each and every department, the extraction procedure totally different. So according to your wish, so what you need for a question, please ask me. So based on the applications, according to the department, Our department means Siddha department, Ayurvedic department, Unani department, different, same plant, different extraction method. Same plant, different mode of preparation method. Same plant, different purification method. Same plant, and that is the extraction method. But basically, the plant pot used for starting material, solvent used for extraction. How many solvent are used for the currently? How many solvent are used to more yield of extractions? So polar solvent, non-polar solvent, dipole solvent. So polar, highly polar solvent, water solvent. The methanol solvent, ethanol solvent. Dipole is a chloroform, acetone, ethyl acetate. Non-polar solvent, benzene and petroleum ether. So these all are the solvent system for preliminary phytochemistry and major phytochemistry. So the preliminary phytochemistry, whether it is alkaloid is present or flavonoid is present or tannin content is present or total phenolic content present, that is a preliminary phytochemistry. Don't go for further secondary metabolites for major phytochemistry, for chromatographic analysis. So based on that solvent system, that is a solvent system. So according to the solvent system and the sample weight, then go for further the purification of extraction of the compound. So extraction procedure according to the department, it will vary the extraction procedure. The choice of solvent, I said the low toxicity. For water is high polar content. நீரில் கரையாது எதுவுமே கிடையாது. ஸோ ஆனால் அப்போ சர்டன் காம்பவுண்ட் வந்து நான் போலார் சால்வெண்ட், பெட்ரோலியம் ஈத்தர்ல தான் நிறைய கரையும் சில சால்வெண்ட். இப்போ என்னதான் ஃபில்லாந்தின் காம்பவுண்ட் வந்து பெட்ரோலியம் ஈத்தர் தான். So depending on the material, depending on the application of the bioactive compound from the plant source, we choose the polar solvent, dipole solvent and non-polar solvent. So easy the evaporation of the low heat and promotion of the rapid physiological and absorption of the extract, so, and the preservation of actions. Okay, according to the solvent used for active compound extraction, water, I said the ethanol, methanol, chloroform, ether, acetone. The remaining two solvent, I said non-polar solvent is a petroleum ether and benzene. Those two solvent non-polar, chloroform, ether, acetone, diethyl ether, these all are the dipole moment. So water, ethanol, methanol, these are highly polar solvent. First three is a highly polar solvent, water, methanol, ethanol. So chloroform, ether, acetone, ethyl acetate, these all are the dipole solvent. The another one is non-polar solvent, petroleum ether and benzene. So based on this solvent system, we are extract the chemical from the medicinal plants. Medicinal plants, whole plant, dry conditioner, medicinal plant in extraction, oven, room temperature dry conditioner, so oven 40-degree dry conditioner, or fresh material are different parts of the plant. So and ratio 5 gram in 10 ml methanol, high polar solvent, non-polar solvent, dipole solvent. So and ratio according to the sample material, then solvent system. That is the ratio versus the sample versus solvent. It is also we have to calculate the general technique of the medicinal plant extraction. There are the plant tissue homogenization, maceration, infusion, percolation, digestion, decoction, Substillator, sonication. But more usually the biotechnology or biological or life science student or faculty members look for the Substillator extraction and sonication. So the remaining method for Ayurvedic and Siddha and Unani and homeopathy preparation for maceration and percolation, digestion, decoction methods. Depends on the industrial purpose or commercial purpose or different department purpose, but mostly scientific purpose or sonication method or Substillators. The 90% or 95% Substillator hard continuous extraction procedure. So in plant tissue homogenations for dry, what I said, dry powder or plant material or wet fresh material, grind it in blade, that is a plant tissue homogenization using mixie. It's a good for shaken vigorously 5 to 10 minutes, then 24 hours. So then extracted filter, then that extract used for the medicinal purpose. That extraction for further human disease or to cure the human disease. So maceration for 3 to 4 days, soak in solvent, different solvent system. What I said, the polar and non-polar and dipole solvent system. Soaking the material up to 3 days, 1 week, depends on the need or applications. So the maceration extraction is prolonged to time up to 5 days, 1 week. Silala fermentation method for maceration, அப்படியே extract எடுத்துக்கிட்டே இருக்கும். Then solvent system based on that solvent good for health. So according to the extraction procedure, definitely the application is good for health. So and the infusion for it is a fresh infusion were prepared by the macerating the crude drug for short period of time with the cold and boiling water, and these are dilute solution, the readily soluble constituent crude drugs. இதை சிம்பிளா சொல்லணும்னா எப்படிலாம் நம்ம டீயை வடிகட்டுறோமோ இல்லைன்னா நம்ம ஒரு கஷாயம் ஒரு ஒரு டிகாஷன் போடுறோமோ அதே மாதிரி தான் டிகாஷன்ஸ், இன்ஃபியூஷன், all those things, digestions, different mode of microwave digestions or traditional stove digestions or stove digestions, depending on the plant material and source of the material and solvent system and the temperature and the prolonged digestion methods. So the thickness of the plant material powder or whatever it is, so according to the plant size and material, then go for the used for the moderately elevated, the temperature is not objectionable. The solvent efficacy on them and thereby increasing. So depending on decoction நிறைய இந்த டிவிலாம் பார்த்துப்பேன் எப்படி டிகாஷன்ஸ் எடுக்கிறது, டீ டிகாஷன் மாதிரி தான், it is a heat salt. Depending on the application, these all are application for Ayurvedic, Siddha and Unani. Maybe or may not be scientific method. And percolation also the same thing. So come to the Substillator. Substillator is a hot air continuous extraction. The fine powder or coarse powder, the crude drug, it is placed in the porous bag. The thimble, I will show the diagrammatic representative for the Substillator apparatus, made strong filter paper, ordinary filter paper, which is placed in the Substillator apparatus. So that is this thimble. The extracting solvent is flask, either polar solvent or dipole solvent or non-polar solvent based on the solvent according to literature or according to the plant material, according to the primary phytochemical chemistry. Go for extraction solvent flask heated and the vapor condensed in the condenser. The condensed extract that into the thimble containing the crude drug. So this is a Substillator apparatus. This is a condenser. The top one is the condenser. So it is below one is the inlet to the water, the outlet of the water. So the middle portion is the extractor. That is a Substillator extractor. The extractor thimble, that is the thimble means this one is a thimble. This one is a thimble. You know, if you know my cursor, so thimble upper part is a condenser. So in bottom of the thimble, that is a round bottom flask, it is extracting or boiling the fresh and extraction of the solvent system. It is a heating mandel, for heating mandel. Are you able to see this diagram?
Yes, sir. Visible.
Okay. Yeah, visible. Okay. It is a 4 different round bottom flask. The round bottom flask attached to the extraction unit or thimble extraction unit. So the symphone amp is there. So after here we are setting the heat according to for methanol 65 degree, for water 100 degree. So for petroleum ether, depends on the temperature, maybe 45 degree or 50 degree according to the temperature. Using solvent system, then boiling the solvent at the bottom of the round bottom, then extract and the thimble going for that way and condensed. So the upper part is a condenser. Inlet water, outlet water. The entire circulation is up to the 1 day, 2 days, 3 days. So 24 hours, 48 hours, then 36 hours. depending on our necessity and extraction, and depending on the solvent, depending on the continuous extraction, this Solstillator apparatus. So go for further studies for the extraction procedure for plant material, plant different parts of the plant material, or whole plant, or drying condition, or wet conditions, or depending on the plant sample material. So we have to analysis. It is not single day, it is not a 1 day parameter. Maybe it will take for 1 year. What I said, all those parameters to execute and plan it for extraction procedure through the Solstillator apparatus. Finally, we are getting yield of the 10% of the extraction or 15% of the extraction from the Solstillator. So that extract go for the animal studies and the case studies after extraction. So the rota evaporate, they recover the same solvent. The yield of the extract paste is coming out. That is the yield of the extract. That extract is a different concentration of MIC, minimum inhibitory concentration, or go for further chromatography techniques for column chromatography, isolation purification, single compound, or any health benefited compound for chromatography, partial the column chromatography, then HPLC, then all chromatography techniques, NMR study for novel compound. Novel compound, if you find the NMR studies are mass spectrum, finally that compound go for the animal studies or cancer study, anti-cancer study. This novel compound is anti-cancer activities there from so-and-so plant, so-and-so extraction methodology, so-and-so compound purification through chromatography method. So all those methods were from the extraction unit, Solstillator extraction unit. The ultrasonication, it is a sonicator, that's a minimum 20 kilohertz to 2000 kilohertz based on the cell rupture of the cells. Some cells, it won't maceration, decoction, and continuous of the hot Solstillator apparatus, it won't separate from the plant material. Certain active ingredient compound from the cell breakdown, that is a 20 kilohertz or 2000 kilohertz, that is ultrasonication method. They come out the such a small molecule active ingredient from the plant material. It is also application is more. So that is a sonication method. So different effect of extraction phytochemical depends on the degree of processing and moisture content and particle size. So these are the plant material. Then for effect of extraction plant phytochemical depends on that. So extraction method, I said these all are the picture. It shows us this is a Solstillator, this is a round bottom, that is a condenser. Same procedure. It is a heating mandel. Depending on the solvent system, depending on the temperature of the solvent system, and the continuous of the or 24 hours or 8 hours, minimum 8 hours, maximum 2 days, 3 days for continuously. Water circulation also illana burst idum. So continuously the length of the period of extraction procedure. Up to 1 week the solvent used, enna solvent use pandrom. And the pH of the solvent is a mandatory. It's a negative logarithm of hydrogen ion concentration of the solvent is very, very important. And temperature also based on the solvent temperature we can choose it. And according to the plant, the particle size of the plant tissues and solvent to sample ratio, that is already I said. So these all are the parameters, the variation in extraction methods also. So before the major phytochemistry, we need to the qualitative analysis. The phytochemical test is a preliminary phytochemical test. Test for drag and drop test or Wagner test, Mayer test, those three different tests, different chemicals, is a phytochemical. Alkaloid is present or not. This is number 12th year BSc lab. So the prominent yellow color precipitation or reddish brown precipitation. So alkaloid is present. Go for further TLC, thin layer chromatography, or extraction, Solstillator extraction, or go for column chromatography extractions or purification of compound. So null compound, if you go for NMR or mass spectrum, bioanalytical technique, finally that compound application is very more important. We can try for all antimicrobial activity, antifungal activity, antibacterial activity. Nowadays, a big issue for MDR, multi-drug resistance. The multi-drug resistance is allopathy drug is more than ampicillin, amoxicillin, rampamycin, azithromycin, all the antibiotic drug resistant. Some corona also resistant. So that is a multi-drug resistance, big challenging for clinical pathogens right now. So we need alternate drug for the alkaloid, flavonoid. It is a curable, easy, the multi-drug resistant disease. It's a curable alternate drug in plant source. The literature is available and also lively is going on. The multi-drug resistant nowadays is a big issue for all clinical trial and human being and the world also. The American Microbiology Society, American Biochemistry Society, all are big challenging for multi-drug resistant. We need for alternate drug from plant source. So that time we are going to alkaloid test, flavonoid test, tannin test, phenolic components, saponin content based on different tests, whether it is present or not. Go for further procedure for major phytochemistry. That purpose only phytochemical screening method. The continuation turbinate, carbohydrate, protein, and glycosides. Different tests, Salcolis test and Moyes test, whether it is present or not. It's already is a 12th practical class or first year's biochemistry practical or any technical institute also there. These all are the preliminary phytochemistry, whether it is present or not, different chemical and test. Then quantitative and total phenolic content for foliage and method, assay method is Singleton and Rossi, 1965. This method is currently we are using the UV visible spectrophotometer. So using the 765 nanometer, it is a visible range of the UV visible spectrophotometer. It is a visible range for gallic acid, for ascorbic acid, or caffeic acid. These are the total phenolic, or rutin, or quercetin. These all are the phenolic compound from the plant source because these all are the health benefited chemicals or phytochemicals. And also the total phenolic content, total alkaloid content, that is a quantitative analysis. So again, the flow chart showing us the extraction of the medicinal plants. So the shade dried and grinded into the powder, and here the sample weight and the volume, then mixed and different depending on the temperature and cool filtration, muslin cloth or any filter paper or any tea filter or what is the moisten cloth, centrifuge at 10 minutes, then separate the filter paper, then go for evaporated 90-degree C. This is a rotavac. Recover the same solvent and the என்ன solvent use பண்ணி recover the same solvent. Methanol recover the same solvent. அந்த solvent திரும்ப same material use பண்ணலாம் up to 3 times. இது rotavac. It's a recover the same solvent. After extracted the bottom of the flask, recover the same solvent. Then solid extract collected the weight, then store the sample bottle and refrigerator. So the extract is stored at a refrigerator for further applications. So this is a plant extraction. Again, they repeated the same slide. Then quantitative analysis. See here the yield of the extract, the hexane, very less yield, 10%. Acetone, 20%. Methanol, 30%. Water, 28%. So the y-axis represented the yield of extraction. The x-axis represented for different solvent system. Which solvent is more yield of extract? So among the 4 different solvent, methanol is a high yield of the solvent. Based on the initial weight of the powder, 50 gram, 50 gram, 50 gram, 50 gram. Different solvent system. The final weight of the powder, then final crude extract, the percentage of the crude, then finally the color of the extraction, dark brown and dark green and etc. So these all are the parameters to find out the extractions. So similarly, the phytochemical analysis, whole aerial plant extract, the Lucas indica. So different test, left hand side different test and the right hand side extraction, hexane, the acetone, methanol extract. The plus for the it is present, minus is it is not present. So similarly, the continuation of the same slide, Then methodology for application for after extraction. I simplify. I never talk about the after extraction. Go for chromatography technique. So now I'm talking about sample collection and process, the methodology for test microorganisms, standardization of test organism, the preparation of plant extract, and reconstitution of the plant extract. It means a minimum inhibitory concentration. After extraction or chromatography technique, the quantitative analysis of compound from the medicinal plant, we are going for the choose the human pathogens or animal pathogen or plant pathogen, any pathogens as a biochemistry student, you are designing and assigning for the minimum inhibitory concentration. The constitution of the extract were reconstituted using the distilled water to obtain 100 mg per ml, 50 mg per ml, 25 mg per ml, 12.5 mg or 6.25 mg per ml concentration. Different concentration is it. That is why we find out the minimum inhibitory concentration using the in vitro study, Petri plate study for different organisms, bacterial or fungal or whatever it is. So that is the minimum inhibitory concentration. Antimicrobial activity, minimum inhibitory concentration, minimum bacterial and fungicidal concentration, and the phytochemical quantitative screening, the crude sample and extract the outcome of the presence of the phytochemical alkaloid, steroid, and tannin, and etc. So, are you follow that?
Yes, sir.
Dear participant, okay. So then, one or two slides going to finish it. So, antimicrobial activity of plant extract, various concentration. Here we are going to talk about 100 mg per ml concentration, zone of inhibition in millimeter. It is aqueous extract, same concentration, phenol, ethanol extract, methanol extract, high polar, polar solvent, different organisms. So the maximum inhibition is 15 millimeter, Streptococcus and pyrogenes. So according to the different zone, that is a 100 mg per ml, the high concentration. Next one is 50 mg. Next one 25 mg. So next one is according there's 100 mg, 50 mg, 25 mg, 12.5 and half of the amount. Then we find out the minimum inhibitory concentration. The first one is the minimum inhibitory concentration nil. So growth all are present. எல்லாத்துலயுமே இருக்கு. இதுல பார்த்தீங்கன்னா மேக்ஸிமம் இன்ஹிபிட் 100 mg ல இருக்கு. இதுல பார்த்தீங்கன்னா 50 mg ல இருக்கு. So the minus symbol is indicate no growth. Plus symbol is indicate growth is present. இதெல்லாம் human pathogens. So human disease pathogens. So Pseudomonas aeruginosa, Klebsiella, pneumonia, E. coli, Bacillus subtilis. These are human pathogens, isolated pathogens collected from the IMTEC source, the Chandigarh. So and minimum inhibitory 100 mg, 50 mg. Next slide for minimum inhibitory is 12.5 mg. So it is a ethanol extract. The previous one is a methanol extract. This one is a ethanol extract. Is a methanol extract. See the 12 mg minimum inhibitory. Plus also growth is present. No growth minus indicate that. So we find out the minimum inhibitory concentration according to the extraction yield from the plant source. So it is ethanol nil growth according to the minimum bacterial site concentration aqueous ethanol methanol extract. This is for different isolates and different solvent extract, same plant and minimum inhibitory concentration. The first one is 100 mg, the second one is aqueous 100 mg, 50 mg, 50 mg, 100 mg, 25 mg, 25 mg, depending on the organism and concentration and phytochemical effect. It's a phytochemical effect. So we need the global demand of the medicinal plant. Already they talk about these are the 7% of annual growth from since the 2000 to the 2050. Every day's utility is more. So our US dollars at $3 million. Right now is 2000 is 2000 billion now. So up to the world market is herbal need. So World Health Organization and Food and Agriculture America, Food and Agriculture Organization from America, and other different organization, different country also to enhance focus the demanding of the demanding of the herbal medicinal plant. So everyone need for alternate medicines. So demand of medicinal plant from the health sector, Ayurveda, Siddha, Unani, and homeopathy and the Western. These are then using the number of species also. That's all. So any doubt? This is also the how many the data comes from the National Medicinal Plant Board under the Ministry of Ayush. So the recent report, the number of Indian medicinal plant 1,742 and the number of phytochemical 9,596 phytochemicals. So it is single plant, different plant, or according to the number of associated. These are the statistics. So if you have the time, please you go through the National Medicinal Plant Board, State Medicinal Plant Board. Those board is indicate, and also the CMAB, I said Central Institute of Medicine and Aromatic Plant Science, Lucknow, and NBRI, National Botanical Research Institute in Lucknow. Those institute also you get the all data bank for the how many people are working in the how many plants according to the medicinal plant and according to the number of phytochemicals. Then that you extract the data from the different source. Then where is your idea and your aim of the work designing based on the existing work. Then the medicinal plant, that is a fact sheet, that is a mega, and how many people are using. So out of 17,000 to 18,000, more or less. So how many plants are utilizing the herbs, shrub, trees, climbers, all those medicinal plant, different forest, different garden, different source of material. So how to utilize that is called the medicinal plant fact sheets. That's all. Any questions, any query regarding the today talk?
Thank
Feel
you.
free to ask.
Thank you very much.
Thank you. Thank you,
very
your
much,
patient
sir.
mind.
Thank you very much, dear participants. Now we are posting the feedback link for the participants. Dear participants, please fill all the columns of feedback link to receive a certificate. Next, that is there any questions from the participants? Please, you can ask 1 or 2 questions because there is no time at all. Dear participants, is there any questions?
Sir,
Yes.
sir, I want to ask one question, sir. So in India, traditionally we are having so much of medicinal
Please,
plants.
little bit loud voice.
1 minute, sir. Sorry, in India, traditionally we have so many medicinal plants, sir. And we are doing so much of research in that area. But practically, the output of the research, so we are finding more novel compounds. But while coming to the drug, it is not coming to the market. What is the reason, sir?
So we need for stage 1, stage 2, stage 3 for application part. First, you have to publish minimum impact factor for journal, reputed journal in the world.
Yes, sir.
So, or go for patent level. Or without publication, go for patent level. That patent going to for in vitro study, in vivo study, and animal studies. Then go for the chimpanzee or a human being clinical study. Then finally go for the market. So up to the publications or patent, then forget about that field of interest. What you said, the novel compound or discovery compound, it won't further proceed. So after getting the young people are looking for the job also. Then go for career, again the family, again then go for that aspect also, not for focusing the same area, same particular area. Am I correct?
Yes, sir, sure. And another one more thing, sir. And also, we are concentrating more on research, but we are not concentrating patent side. So this is also one of the drawback.
Yeah, drawback. That is India is a more drawback.
Yes, sir, that's a problem. Because we are doing research, but we are not patenting.
Yes.
Hello?
Yes, sir. Thank you, sir. Thank you so much, sir.
Ask. No problem.
Sir, another one more question, sir. During the extraction process, we are using aqueous extract and methanolic extract and methanolic extract. But in the same time, most of the compounds are extracted in the methanol extract. So is there any reason for...
So high polar solvent is a water, methanol, ethanol, I said. And
Yes,
also the low toxicity methanol and ethanol. Compare than other solvent system.
Yes, sir.
Low toxicity. So we need
okay,
to the 100% the methanol clear through the rotavac or evaporate 1 or 2 days the open air condition, then remove the complete ethanol and methanol, but least percentage of methanol effect or ethanol effect doesn't bother our, that is the least effect. So that purpose only every one of the scientists is looking for the high polar solvent content, either water or methanol or ethanol, then the remaining solvent, dipole solvent and non-polar solvent system.
Okay.
Thank you, sir. Thank you so much, sir.
Now the feedback link, please put it in the chat box.
Okay, sir.
Any other
questions from the participants?
question?
Already, Sir has delivered a speech for
Actually,
more
you
than
gave
1
a
hour of
elaborated discussion, sir.
The speech is very
Literally explained.
useful, which is very useful to each and every participant. Any other questions from the participant side? If there is no, we'll go for the vote of thanks.
No, if you are interested, my humble request, sorry for the disturbance. Okay. So
No,
if you are interested for a bioanalytical technique for chromatography technique, it is take for 1 hour per talk.
Surely, sir. We will arrange it, sir. Surely we will arrange it, sir.
We will arrange it for next webinar, sir. Actually.
Yeah. If you are interested for NMR processor or out of a novel compound, then go for patent or any.
Sir.
Yeah.
Yes, Rajesh, put to the link. Link is not available. That is feedback link. That is, dear participants, kindly fill this feedback link to receive the participants. So there is the concluding part of our today's webinar. Dr. A.Annamalai, he has covered each and every part of this today's presentation or deliberation. He delivered a talk on medicinal plants, not only medicinal plants, how to use the plants and the importance of plants and how to do the research on various plants also. And one more thing he has quoted, which is very suitable for the present generation, that is Unave Marundu. That is, food is our medicine. Unave Marundu. He rightly pointed out this point. And this youngsters and young scholars and young students and faculties, we should carry out the research work. Not only research work, we also get the patent also. As Sir said, not only to do the research work on various medicinal plants, but also we have to get the patent right. Each and every medicinal plants, here, Sir clearly pointed out that is Philanthus Amaris. It is very good medicine. It is available only in India, not in other foreign countries, right? So Amaris, that is Philanthus Amaris, both Annamalai sir and myself discussed many things on this Philanthus Amaris during our Karunya University life. And sir did excellent work on Philanthus Amaris, and moreover, many of us use the term that is Philanthus neruri instead of Philanthus Amaris. So it is a new thing, and it's very informative session, sir. Moreover, sir has elaborated the use of medicinal plants for various ailments, for obesity, cancer, diabetes, ulcer, and also we can use it for anti-aging purpose also. What not? Not only that, that is, yesterday I spoke to Dr. Annamalai sir to restrict the talk for 1 hour. But he took more than 1.5 hours, nearly about 2 hours. It shows the keen interest of Dr. A.Annamalai sir, and moreover it shows his interest towards the scholars and also to the society, young society. So he elaborated a very good talk on research part also. And next time, that is, if time permits, and we request Dr. A.Annamalai sir to take a webinar on that is phytochemical isolation and also extraction procedure in a elaborated manner.
Sorry, yes.
And
Phytochemical is a live talk is very important, sir, than a webinar on the digital mode.
Actually, this digital mode also it is reachable now. Moreover, participants, nearly about 100 participants, it is still maintaining in our webinar. It's a fascinating thing, and moreover, it is very useful and informative. And all the participants, they were eagerly listening your talk, useful talk. And we are all
The session is
on behalf of the Department of Biochemistry and all of our participants in and around Tamil Nadu, that is, we are very eager to listen your talk. But we are also cooperating with your talk, but our network is not cooperating with us. So that is nearly only 5 or 10 participants left from the meeting. But it is immaterial. So many participants, they have given a good feedback on your presentation.
Thank you.
Very useful talk, sir. And
சொல்லுங்க சார்.
சார்,
எக்ஸ்கியூஸ் மீ, அவருக்கு ஏதோ ப்ராப்ளம்
இருக்குங்க சார்.
ஓகே
சார், தேங்க்யூ ஸோ மச் சார். பிகாஸ் வந்து நிறைய இன்ஃபர்மேஷன்ஸ் வந்து உங்ககிட்ட இருந்து கேதர் பண்ணியிருக்கோம் சார். எல்லாரும் மெடிசனல் பிளான்ட்ஸ்ல ஒர்க் பண்ணியிருக்காங்க, பட் வந்தாலும் எலாபரேட்டடா நிறைய ஒர்க் வந்து நீங்க பண்ணிருக்கீங்க. ஸோ யூ டிரான்ஸ்ஃபார்ம் த மெனி இன்ஃபர்மேஷன்ஸ் வித் அஸ் சார். So you spend very valuable time, because 2 hours அப்படிங்கிறது it is impossible, but without any hesitation and all these things you spend lot of time with us, sir. Thank you so much. Once again we arrange another one more meeting, sir, for explain about the bioanalytical techniques, because it is very important, because I already told that is NMR, and because everyone knows about NMR, but how to know the results and how to interpret the
interpretations.
That's very important, sir, because we are lacking on that part. So definitely we will arrange. Thank you so much, sir.
Thank you.
Dear participants,
shall I move?
Sir? Okay, sir. Okay. Sir again come or not? Sir left the meeting.
Left the meeting because of the network problem. God's grace, no network problem here.
Actually எனக்குமே நெட்வொர்க் கட் ஆயிருச்சு சார்.
Okay.
But ana continue
Fibernet is good, sir.
Sir,
Fibernet is good.
Yes sir. Sir,
okay, shall I move?
Sure sir. I think
okay,
sir will make a call to you, sir.
Okay
sir. Thank you so much. And dear participants, really thank you so much for your constant support and participation. Thank you so much.
Okay.
Thank you, sir.